Hi, good afternoon. I'm Mohammed Gassimi, and I'm an assistant professor of computer science at Michigan State University and a national scholar for data and technology advancement at the National Institutes of Health this year. I'm joined today by my colleagues at the NIH, Dr. Grace Fang, Drs. Jim Knott, Christina Fang, and Susan Wright. And today, we have had a slight technical issue, which is going to change some of the format today. We will not be having a debate because of some technical challenges, but we will instead be performing an interview of Professor David Kleinfeld, who I'm going to introduce momentarily. Before I kick off the interview, I would like to emphasize 2 points. The first point is that in the course of this debate, Professor Kleinfeld has very generously offered to cover the two sides, sort of, the arguments for and against our debate question, which is, can we generate an organized theory of the brain? And so I want to really thank Professor Kleinfeld for agreeing to sort of help us understand the nuances in the for as well as the against side of that discussion. The second thing I'd like to bring up here at the start is that we're very interested in hearing from members of the audience with your thoughts, reactions, and follow-up questions. So please go ahead and post those in the questions window, and we'll just sort of pass those along when the interview period concludes and we enter the Q&A period at the half-hour mark. Okay, so with that preface out of the way, I'd like to briefly motivate the topic for today's interview. The human brain is a mysterious organ that translates electrochemical impulses, neurons, into thoughts, feelings, and behaviors. And so there's some hope that if we could organize and understand the activity of the human brain, we might be able to harness that knowledge to treat brain disorders such as Alzheimer's or brain abnormalities like autism. Or maybe we could even get some insight into great unanswered questions of science, like what is consciousness? And if you listen to some of the talks earlier today, you've probably seen that neuroscientists are making great progress towards unraveling some of the mysteries of the brain, but we still are not or don't have in our clutches a single, unified, organized understanding of how the brain works that's akin to, for instance, our understanding of how transistors work together to enable your laptop. So this is sort of an overview of the motivation or the motivating question for the debate, which is, can we generate an organized theory of the brain? Now, having said that, I'd like to very quickly introduce our distinguished speaker today.
That's Mohammed. Yes.
Mohammed, I think Florian is actually on the phone.
Oh, wonderful. Okay. So okay, that's great. So what we're going to do is we're going to come back to, instead of structuring this as an interview, we're going to kick it back then to the original debate format. So I will introduce both of our distinguished speakers. And I want to note here up front that I could occupy this entire session reading off their many accomplishments. But given that we had a little bit of a delay, I'm going to give you the abridged version of their backgrounds. So representing the for camp for the purpose of the discussion today is Professor David Kleinfeld. He's a distinguished professor at UC San Diego, where he leads a diverse, multidisciplinary laboratory that focuses on issues in systems neuroscience. He's a member of the American Academy of Arts and Sciences and a fellow at the American Association for the Advancement of Science. Dr. Kleinfeld, thank you for joining us. And we also are joined on the line today by, who is representing the against camp, is Professor Florian Engert. He's a professor at Harvard University, where he leads a laboratory studying neuroethology and the evolution of adaptive natural behaviors. He's a recipient of several awards, including awards from the McKnight Foundation, the NIH Director's Pioneer Award Program, and the Human Frontier Science Program. Dr. Engert, thank you so much for joining us.
It's a pleasure. Welcome. And thank you for having me.
Absolutely. So, okay, having said that, I've given you an abridged version of these two giants' backgrounds. I'm not going to be able to mention all the other wonderful accomplishments they have, so let's just jump into the debate now. We're going to begin with some opening statements from each side. I think 5 or so minutes is what you could aim for, but you can go a little more, a little less as it works for you. And then we will pose additional questions thereafter. So let's start with the for camp. Dr. Kleinfeld, I'd love to hear your thoughts on what evidence is there to suggest that we might one day be able to generate an organized theory of the brain.
Okay. I mean, some of this, of course, is based on hope, but you might say I started out in physics, and you only go into physics if you have a certain hope that you'll have a somewhat simplified description of the world. But my feeling is this: we have a pretty good understanding of dynamical systems, both at the low level, which means we could write down fairly exact so-called conductance-based equations for single cells or groups of single cells, put in a lot of what we know about the kinetics and thermodynamics of channels, and get a very good model representation of what we see, and maybe more importantly, we even have a lot of knowledge about how to abstract this. And we also have a good knowledge of dynamical systems at the very large level, at the population level. It doesn't mean we're averaging out all the details of when different neurons are active, but it allows us, again, to have a somewhat simplified view, a conceptual understanding of what's going on. And there's been great strides. I mean, everybody has their favorite experiment. My favorite experiment is the ring attractor that Vivek Jayaman identified in the fly, that Chaim Sampolinsky worked on theoretically in terms of a stat-mech model. But it's a great achievement, but on the other hand, it's really a one-dimensional system. And I think what's missing is there's a serious experimental understanding of the dynamics of systems where the same elements which are imposing the dynamics, synapses, somata, are also storing information and being continually modified. And this is quite different than the situation we have in electronics. And we know a fair amount about synapses on the one hand. We know about things like spike timing dependent plasticity, so the work from study with Curtis Bell and then Mu-Ming Pu. But we really know very, very little about plasticity. It seems to be the one area that's neglected. John Nye was talking this morning about the cell census and all the details about neurons and their development and maybe how they're hooked up together, but we don't understand, we don't have experimental clues to allow us to develop a theory which includes plasticity at the connection between neurons and to some extent in the expression of threshold levels of neurons themselves. So I'm optimistic, but I think there's a big hole in our gap, and there's a dearth of experimental evidence. And I must say, I'm just thought the sort of the excuse of this debate with my friend Florian is a good point to represent this hole in our knowledge, because it's also, I think, the kind of program that the brain effort should go after. It should go after fundamental holes in our knowledge. Okay, so that's that you may say is my treatise. Optimistic, but we need some strong experimental clues to understand the distributed memory and plasticity within the nervous system.
Awesome. Thank you so much for your opening statement, Dr. Kleinfeld. Let's continue now with an opening statement from Dr. Engert, who has kindly agreed to sort of serve in the against camp. I want to emphasize again here that all scientists, I think, hold nuanced views on complex questions. So it would be unfair to say that either Dr. Kleinfeld or Dr. Engert are strongly representative of either a for or against camp. But for the purposes of educating us in the debate today, they're agreeing to sort of highlight the different sides of the issues. So having said that, Dr. Engert, I would love to hear your thoughts on what is the evidence that suggests that we may not be able to generate an organized theory of the brain.
Yeah. So the question is an organized theory of the brain, and I'm still struggling with what exactly the question is, or what would constitute, or when would we know that we actually have a theory now that we didn't have before. So I think at the heart of everything here is the question of what constitutes understanding, and when we actually think we have manage this leap from ignorance to understanding. And I had a wonderful discussion preemptively for this meeting today with David Kleinfeld 2 days ago, when he called me when I was hanging out in a mountain hut in the Dolomites. Now I'm at the oceanside campground in Italy. So I apologize for the telephone, and I hope my connection will hold. Anyway, I think the two examples that David brought up 2 days ago, where we did not have a theory and then we had it and we knew. One was understanding atoms, and we needed quantum theory to understand them. And I think that's an excellent example, where a theory was missing and then we had it and then we could move forward. And I think the other one was magnetism, where the Coulomb forces, like the repulsion of charges, was not sufficient to explain magnetism. And I think ultimately we needed special relativity to as a theory to explain magnetism. I think these are two excellent examples. Maybe somewhat more stretched one is the theory of evolution that explains the diversification of species. And I think at that level, I'm not sure we are lacking a theory. I think there's everything we need to understand neural networks and dynamical systems. These fundamental principles are already in place. I don't think we even need quantum mechanics to explain the brain or neurons. I also don't think we actually need magnetism. Of course, all of this depends on what you mean by understanding and how deep you have to go. So I think our current misunderstand or a lack of understanding of how the brain works is really based on synapses and neurons and the interplay between them. So my hope is that the current principles we know about neuroscience, mainly about how neurons work in principle and how synapses work in principle, is sufficient to explain the computations that all brains perform in order to solve the solutions that animals need to solve to survive and to be adaptive. So having said that, maybe another analogy is, are we also lacking a theory about the heart or the kidney? Yes. Because I mean, some people say that we have theories or we have an understanding of how electronics or man-made objects work, like computers and transistor radios, and but we lack an understanding of biology, because biology is just much more complicated and harder to understand because we haven't built it. I agree with that. I think understanding how a cell works is still beyond us currently, a single cell. But I don't think it's for the lack of a theory. I think it's for the lack of putting together all the little bits and pieces, starting with biochemistry, with physics, with all the mechanisms, the biological mechanisms that explain how cells work at the cellular, at the molecular level, how biochemistry works. I think if we put all of this together, then understanding of how the brain works will slowly emerge. And it's not the lack of a cohesive theory, it's really the lack of putting all the individual bits and pieces together. And I think slowly in the next decades, an understanding of the brain as a complex dynamical system will start to slowly emerge without a fundamental paradigm shift that we are all waiting for, or that's currently under debate. I want to finish my my initial statement with a poll that maybe you can conduct amongst all the audience. And I would like to ask them if you give them a choice between A and B. Yes, so fourth choice, they have to choose either A or B. And A is, we are still lacking a fundamental understanding, a theoretical understanding. And once we have discovered how this works, we will have an insight, a eureka moment, and we can move on. So that's option A. And option B is that we have all the principles in place, and it's just a question of incrementally advancing it. So then I would like to know what the audience, how they would vote, right? Do they think there is some fundamental mystery or some theory still lacking? And once we have found it, we will know and we can move onward at a different level? Or is it just, as I previously suggested, baby steps? I don't know whether it's possible to do this quickly with an audience like that, but I do this frequently when I have an audience at my disposal, and it's actually fascinating what the responses are. It's usually 50-50, and the answers are diverse and very interesting. Anyway, that's what I have to say as an opening statement, and I'll hand over the mic back to Mohammed or David.
Thank you so much, Professor Engert, for the opening statement. So one thing that I heard echoed by both of you was I bring my own bias as a computer scientist to wanting to draw an analogy between the brain and obviously moving from a transistor system all the way up to a computer that has an operating system that goes on top of it and has all these layers of abstraction that we can understand how each of those work together to inform the other. And I heard from both of you in the course of your opening statements that analogy has flaws. What I was hoping that we could do is expand maybe on each of your perspectives, or from each of your perspectives, on if there is a good analogy to think about, or if the system is unanalogous. There's not a good sort of parallel for people to think about when they want to understand what a unified theory would look like. Because in the computational sense, we think if we can understand from the transistor all the way up to the OS, then we've got it all pinned down. What is the right way for us to be thinking about this when we want to speak about an organized theory of the brain, if the transistor sort of analogy is not the right one? Maybe we can start with Professor Kleinfeld, and then we can move to Professor Engert thereafter.
Okay, so I mean, as you point out, Mohammed, in electronics, you can write down all the equations. You could even simulate them with all their ugliness in programs like Spice. But again, the one thing that's special here is that the devices themselves have reliable properties, predictable properties. I mean, they may not be linear. I mean, diodes are voltage-dependent capacitors, et cetera, but this works. And to the extent that you have a nervous system and the components are relatively fixed in time, you can make good predictions. I brought up the recent example of the central complex, the heading sensor in the fly. If we roll back almost 40 years ago, I mean, there was beautiful work on small systems in Tritonia, in the gastric mill in the stomach of the lobster, and even allowing for things like modulators. As long as the level stayed constant, people were able to make good models of the circuits. But I just want to harp back again a little to the issue that the components are plastic, and this is something that's just beginning to come into the electronics world in like so-called memory devices or memory resistors. But I disagree with Florian that we know enough. I think we don't know enough. I think we barely understand memory. We have an idea of eligibility traces as a concept to extend correlations across events in time when we take the events one at a time. But we don't see the world one at a time. We see the world as many events at a time. So there's many eligibility traces, just to pick up on a theme. I think we're extremely naive about issues of plasticity, and there we need experimental guidance. I also just, I mean, not to harp on physics, but it provides good examples. To go back to the issue of magnetism, I mean, what Florian said is true. Coulomb's law of special relativity allows you to calculate the residual effect when you have balancing charges. This gives you magnetism. But to understand magnetic materials, you often appeal to an intermediate level of understanding. So it's very difficult often to calculate the detailed interactions between magnetic centers in a material, but you know enough about symmetries that have to be obeyed that you could put in by hand an interaction which is physically reasonable and then calculate all the way up to, say, a macroscopic quantity. And I think science, at least physics, chemistry, has made progress by putting in intermediate steps and then only later justifying them in a microscopic way, as has happened in superconductivity and magnetism as two famous examples. So just for the sake of the argument, to disagree a bit with Florian, who I love dearly, you, I mean, I think
Hey,
you
Mohammed,
can't think
can
of,
we interrupt each other? Or
you always interrupt me, so that's okay. But
no. I'm just wondering whether what's the format here, whether we can
Uh,
turn this
uh,
into a discussion or whether you
yes,
prefer to have these separate statements.
yes, I think so. From my perspective, I think a discussion back and forth, is fine by me. So,
Okay. Because
okay,
what David just said is something that I actually, I mean, I almost agree with, right? I think David is absolutely correct that the rules of plasticity, I think, is something that should be almost discussed separately, because there's sort of one question about how the brain works in terms of processing information. The other one is how it works in terms of memory and learning and plasticity. And David already in our previous discussion 2 days ago pointed out that that's where the analogy falls short with computers, because computers are not plastic; they can't update them on the fly, and I think that's absolutely true. I think the only thing where David and I disagree, maybe, I'm not even sure we fundamentally disagree, but I can generate an artificial disagreement here for the sake of discussion, where David thinks that the plasticity rules that we have discovered so far are probably not sufficient to explain the dynamic plasticity of the brain. And I will, for the sake of argument, put forth here that they might be. Yes, that the rules of plasticity that we have discovered in the past, which is spike timing dependent plasticity, which is reinforcement kind of plasticity, that sort of addition rule of dopamine or serotonin of neuromodulators that can change synapses, that at least in my mind, these rules in principle could suffice to explain, I think, all kinds of learning and memory in theory. Yes. Like the eligibility traces that David mentioned before, I think they can just operate in parallel in a complex neural network like the vertebrate brain. Where I actually agree with David is that it's likely that we have fallen short in describing all the complexity and plasticity of the vertebrate brain. It's probably a very good idea, and I'm totally on board with David, to systematically test these fundamental plasticity rules experimentally and find out whether my somewhat naive assumption that spike timing dependent plasticity and reinforcement rules might be enough to explain it all. Because most likely, if we actually do these experiments and test what plasticity rules actually exist at the cellular level, I'll be proven wrong, and David is going to be proven right. So back to you, David.
So I actually want to ask a follow-up that ties very nicely, I think, into this, which is, there is obviously some differences of opinion, some of which I think, as Florian alluded to, is performative because, scientists hold nuanced opinions. But continuing along that strain, there is a difference ostensibly because, or perhaps there's a difference because of some experiments that have not yet been run, or an inability to collect, let's say, a certain kind of data that would allow you to get rid of a hypothesis, either prove it correct or incorrect, and so on. I'd love to hear from both Professor Kleinfeld as well as Professor Engert about what are those experiments, what kind of tools would be necessary to help get to an authoritative answer to close out this debate, and are those on the horizon, those kinds of experiments? So I'd love to, maybe we can start again with Professor Kleinfeld and then move to Professor Engert.
yeah. So even though what we're doing very now is really sort of macroscopic systems neuroscience, I really think that the field could take a shift back to a reductionist view, and particularly so when it comes to issues of plasticity. And let me just give 2 examples where cell culture was key. I mean, one, of course, is spike timing dependent plasticity. This was our colleague Gao-Chung B's work with moving poo. This is a culture experiment. There's always arguments, oh, it's in culture, it's not the same as in the animal. But to establish fundamental principles, I think the ability to go into the dish, the ability to resolve individual synapses now. You know, you could to image at quarter micron resolution. Just push the technology, some of which came out of the brain program, is the way to be. And another famous story that came out of the dish is our scaling laws. This is Alex Ray's experiment. It was, to me, it was sort of a shocking finding. There was a theory about balance between inhibition and excitation currents into a cell. So the mean is close to zero, but the cell is very noisy because the variance is adding. And in order for this to happen, you needed a kind of scaling with synaptic density that went like the square root of the density. I mean, Alex found something very close. So, I mean, to push this further and understand, is it spiking in the postsynaptic cell, or is it just depolarization, or what the molecular events are, as a few people in the world are doing beautifully? I think this needs to get pushed. I think we need a very reductionist view towards pushing towards synaptic physiology. And the flip side is there are good tools now to look at individual synapses within the same cell. You know, I'm thinking of glutamate dyes, calcium dyes that are localized to a synapse. We could get an idea of what the correlation lengths are in individual cells. I mean, people still fight about this. Or is the spine isolated? Is the spine not isolated? How far apart are synapses that influence each other? I view these as basic questions, and I think the technology is pushing there. And you know, again, I'll make a plug for the brain program. The ability to push all these technologies, dye makers and physiologists and optics jocks in the same direction has been great, and it's still growing. I mean, there's no end in sight. So I think there's basic experiments looking at the microscopic level, both in the animal and in the dish. So Florian, what do you think?
I think that's great. I think I'm all for the developing of technology, because I think even if I don't necessarily agree that we need these experiments, say at the single spine level, whether the spine is isolated or not, I don't think we actually need them to understand the brain at the macroscopic level. I think that's sort of a question here. Nonetheless, if you do the kinds of experiments that David just suggested, you make inevitably important fundamental discoveries about basics that will help you understand the basic biology of the brain. I think this is always useful. My hope even, or my suspicion is that these details don't really matter all that much in terms of understanding the complexity of circuits and behavior. So I think the experiment that Mohammed was referring to, that I would like to do, that to prove my point right, namely that we don't need a further theory, is that if we just take the principles that we have in hand right now about synapses, about excitation, inhibition, modulation, about the basic rule of plasticity that we have in hand, and if we start putting all of these elements together in a small and simple brain, like in my case, for example, the larval zebrafish. The larval zebrafish has 100,000 neurons, and I think in principle it's possible now to model the whole zebrafish brain. My prediction is once we have the data at sufficient resolution, I think what we need in this case is basically a connectome and an insight about the connectivity, the synapses that exist. Once we put all of these existing principles together into a brain like the larval zebrafish's brain, can we run it and generate the output of a larval zebrafish? So can we basically simulate a simple organism like that with all the elements that we have in place already? If that works, I would argue, then it tells us that we don't need to add any more details or a fundamental theory to understand at least the fish's brain. And I think the other experiment, the fundamental experiment I would add, is to start putting together the subcircuits that elicit the individual behaviors of a larval zebrafish, like hunting, like escape, like rheotaxis, optomotor response. And if we can simulate, so in a computer, recreate these neural networks, run them, and then generate an animal in vitro that is indistinguishable from a live animal. I think that's an experiment that sort of tells you that our theoretical framework is sufficient. So I think that's sort of the experiment I'd like to do, to show that we have all the fundamental tools already in hand to explain at least the tiny vertebrate brain, or any brain. And then I would argue that the rest, going from a larval zebrafish to a juvenile, to a mouse, to a monkey, to a human, is just a matter of expanding, right? It's not a fundamental new thing we need. It's just a way of slowly and deliberately expanding our knowledge and the complexity. That's how.
So Florian,
over David, over to you. Yes.
So the thing is that if there's a behavior that a zebrafish does that is quite plastic at learning quickly, like just in one of your fish tanks, as
like
you change
gain control, right? They do motor learning. They do very quick motor learning, gain adaptation.
Okay, so why? But that has periods of learning and periods of stability where it's not learning.
Yeah.
And so how do you? I mean, it always seemed interesting to me, and I would say unresolved, to know when are you actually learning, and when do you have stable output? And you can't say, oh, there's a modulator, because that's a chicken and egg problem.
Yeah.
How do you maintain?
No. It's,
So I see that as fundamental. I see that as a fundamental flaw in our theoretical understanding of nervous systems, is the ability to pick a certain level of stability. So when we get up in the morning, we're the same person, but yet we could learn in single trial learning. I mean, it seems miraculous, but it's miraculous because we really have no theoretical clues, unless I'm missing something.
Yeah, maybe I don't know. I think it could be that it's a question of sensitizing a system such that it's plastic, like a boost of adrenaline that can be elicited by a mismatch signal from an error signal that comes from the world, that can generate a neuromodulator that suddenly induces a brief transient period of plasticity. And if that mismatch error signal that can either come as an unconditioned cue from the outside world, or it can be a mismatch signal between a reafferent stimulus and an efferent copy, that induces brief periods of plasticity, or like a surprise signal from dopamine, right? You have an error prediction signal, or a climbing fiber signal, right? From the inferior olive into the cerebellum, there's a teaching and error signal that will induce brief periods of plasticity. And of course, the question is what induces these brief periods of plasticity, but this can either be an external cue that makes the current context more relevant to the animal. So there's no mystery there. There's no fatal misunderstanding. Or it could be a mismatch signal between your efferent copy, like a forward model that gets generated by the brain, and the reafferent signal that comes back. And if there's a large enough mismatch, then you open a brief window for learning and plasticity. How about that, David?
Well, it sounds good, but I think there's some flaws. I mean, a climbing fiber signal goes to a specific cell. It's an instructional signal that goes to one Purkinje cell, and maybe one could argue that there's some reasonable understanding of the cerebellum, at least within its own right, within its own closed loop. But a dopamine signal goes everywhere, and and I'm not even sure people are even looking in the right place for things
maybe
like correlations. They like the striatum because
I agree, David.
Yeah, so I
agree. I think that people might not be looking at the right place, but I don't think we are lacking a fundamental theory. I think the theory is there. It's just a question of getting people to look in the right place. I agree with that.
Okay, you're a little. I think you're playing devil's advocate here. I mean, I take reinforcement theory to a point, but it's really only considering sort of single learning exercises at a time. It's not trying to learn, multiple. You're not following multiple inputs with multiple solutions like you do during the day. You walk around your lab and
Yeah.
Yell at every
I agree.
postdoc and student individually,
no, I
right?
agree. But I think that's poverty of experimental design or the quality of the experimental design. We are not asking a mouse to learn or a rat to learn many things in parallel. We are actually deliberately constraining our experiments to ask one time. And I think that's the...
Oh, I think we lost you.
Hello?
Yep. Oh, we're here
Am
again.
I back?
Yes.
Yes. So I think that the... Hello?
Dr. Engert, we lost you
Can
again.
Can you?
Yes, we can hear you. Please go ahead.
Can you hear me now?
Yeah.
Yeah, you're sort of off and on.
Okay,
yeah.
Good. Sorry. I think I've got my phone too close to the ear. It presses 0 accidentally. Anyway, I think David's right, but I think that the issue is that we design our experiments such that an animal needs to learn one thing at a time, like a mouse in a classic reinforcement learning experiment, or even a monkey. So we are not designing our experiments to ask whether animals can learn many things in parallel. And I think if we did that, we could redesign experiment to ask exactly these questions about multiple eligibility traces and parallel learning in a rapidly changing world. So I think it's not a problem, again, of a lack of theory. It's a lack of, I wouldn't even say poor experimental design. I think simplifying things when you want to study them is often useful and asked for, especially in physics. And probably in neuroscience, we have wandered off the beaten track, in particular with respect to reinforcement learning in mammals and rodents, where we ask probably mice and rats to solve the wrong tasks. And maybe that's the problem right now, why you can't really make a lot of progress with parallel learning, like dopamine reinforcement learning, is because we are not really asking the mouse the correct question. What do you think, David?
I mean, I agree. I mean, we could, and we would go ahead and do different experiments maybe. I would just take a more reductionist approach, but I'm also, even at this high level, I'm just very nervous about what we learn. There seems to be a lot of groupthink in the area that studies plasticity the most, which is sort of the reinforcement dopamine world. Hopefully, I'm not offending anybody, but one takes risks. But it doesn't. There's a lot of things that, as you point out, seem like contradictions. The place where the information is the densest, which is cortex, or even hippocampus, is not where we're looking for the plasticity effects. Yeah.
I agree. I completely agree with that. I think that there is a lot of improvement, I think, possible for just experimental designs and also how you ask the right question. I agree with that. I don't think that we are actually lacking a fundamental understanding or a theory how it could work. And maybe I should also say something about sort of a dichotomy between understanding. I think there is a lack of like an ignorance or a lack of understanding where we have no idea how it could possibly work. Like if you watch a stage magician, it's just a mystery and we have no idea. And another form of ignorance is where we have many ideas how it could work. We just don't know whether the model we have, our intuition, is actually correct or not. That's a different form of ignorance. And my opinion about neuroscience and brain science, it's that we are more struggling with the latter, that we can come up with ways how it could all work and it all makes sense. We just don't know whether we are actually on the right track. I think, but that's a different form of ignorance. It's not the ignorance where we just puzzled and we are clearly lacking a theory, whereas it's one where we just need to look more carefully and convince ourselves which of our several intuitive models is actually the correct one. Over.
Okay, so I think this is maybe the at least for the sake of this public debate, this is where we part company a little bit. I think we're missing some fundamental experimental key, like looking at the magician from behind the stage or something to see what the trick is. Yeah.
And also,
Okay.
again, I should really agree with David here, is that I think these experiments that David is asking for, I'm also for them. Yes, sir. I think they will reveal new insights that are fundamental, and they are fundamental at the detailed molecular and mechanistic level. And I think they're going to be immensely useful. Yeah. I think maybe the only way where we disagree, I don't think that they are going to be absolutely critical for us to understand the brain at a new and different level. Over.
Okay. If I may, just for the purposes of allowing us to get to the audience interaction section here, I'd like to pivot the conversation to questions that are being posed by members of the audience. And I know that some of the other members from the NIH team had been keeping an eye on some of the questions and had highlighted those for me. So if you give me a moment, I'm going to pull those up on my end. Okay, so I'm going to start. We're going to switch now very quickly to the audience portion of this discussion, and I'm going to open up with asking the first one here, which is, might it be possible to have local theories at different scales of understanding? That's the opening question from the audience.
Okay, I mean, Florian, I'll just sort of mention this. I mean, again, I mean, physics does provide very nice use of this. I mean, we're almost all used to this. I mean, we don't think about quarks. We think of nuclei, and then if we're doing chemistry, we don't even think about the whole atom. We just think about the valence electrons in the atom. So if you're trying to go from atoms to molecules, it's just valence electrons. If you're trying to build materials, you start to think about the molecules as a whole, and then that's a whole nother simplicity. You don't even have to think about details of quantum mechanics. You just think about what is the square of the wave function, just the electron density, famous functional density theory. So I think, again, appealing to physics and chemistry as sort of successful science for the design of structures, it's usually hard enough to just connect two levels. And so you do this in neuroscience, of course. You don't think about channel kinetics. You really think about neurons spiking, and you don't even think about the details of the spike. You just know that there's a probability for synaptic release, say, and you know that there's a probability that cells will fire, and you could read them out. So we're on that level, we're very good at connecting. But I mean, I would bring up just one point here before giving this to Florian, which is a comment that my colleague, Florian's colleague, Chaim Samplinsky brought up, is that the challenge of the brain is that on this sort of hardware level, you have these intermediate steps. From the point of connectivity, you have connections on all possible scales, and that's what makes it difficult. This segues into Florian's previous comments about the connectome. So, Florian, what do you think about this?
Well, I guess the question again was whether we could have theories at different levels. Is this right, Mohammed?
Yeah. Yes.
Okay. So I think that our theory of the neurobiology of neurons is actually almost rather complete, relatively speaking. I think we have a very good idea how neurons work, how receptors work, how channels work. I mean, how the potassium channel opens. We know that. I think we know how synaptic vesicle release works to an extraordinarily satisfactory level. So I don't think that we are actually lacking a theoretical understanding at that molecular level, at the synaptic and at the neuronal level. So I think it's more the question that we are struggling with, that is how this vast dynamical system, this emerging dynamical system of 100,000 or 1 million or 1 billion neurons that interact, how that gives rise to behavior and thought. And so I'm not sure that the theory at the molecular level is lacking or would help us that much. But I don't know. It might actually. Again, I have to say this again and again. I'm just speculating. I'm not talking from a position of certainty in any way. I just try to keep this debate lively. So to answer the question from the audience, I don't think that we are actually lacking a theory at the mechanistic neuronal level. I think the challenge really is just to apply these principles that we know already to the level and scale of neuron and synapses at the magnitude of a vertebrate brain. Over.
That's actually a good transition into one of the questions that was related to this, again from the audience, which I'll read out. So a member of the audience asked, maybe we can phrase the question a little differently. A theory should allow accurate predictions. Can the complex nonlinearities have multiple interdigitating timescales of the brain be simplified into more simple equations that can make interesting, accurate predictions? What level of accuracy would we desire, and what do we want to predict?
Oh, yeah. Can I take that?
Absolutely.
Please. So
yeah, please, go ahead.
I think that the brain is fundamentally a stochastic prediction machine, not an accurate deterministic dynamical system. So accurate predictions, I think, are impossible. The only thing we can hope for are stochastic predictions. And I think that's fundamentally, the reason for that is that the neuronal processes at the fundamental level are stochastic in nature and not deterministic. I think the other problem with the goal of making predictions generally how a brain will behave.
Dr. Engert, I think we lost you.
Oh, I think you lost both of us.
Oh. Can you all hear me okay?
I hear you. Yeah, I hear you, and I don't, somehow the video is dead. But that's okay.
Yeah, I think that's fine. I mean, it seems that Dr. Engert we've lost him from now. So maybe Dr. Kleinfeld, if you want to, we'd love to hear your take on it as well.
You know, on the question with what precision you could define, what's the precision that makes a theory valid?
Right. I'll go ahead and repeat it. So the sort of the core of the question was, can the complex nonlinearities at multiple timescales of the brain be simplified into, more consolidated equations that can help make accurate predictions? And then if that's true, what level of accuracy do we need, and what would we want to predict with that?
Okay, so at one level, you could say the bottom line is really predicting motor output, and what's the chunking in time you need to predict motor output. So we have a little bit of a clue from that, even from industry, for vector quantization, say of voice, which is one great motor output, and we're really chopping voice at something that approximates about 10 hertz into intervals. So I think you could say that for a theory that enables us to compute how we can communicate, you could chop time. I think we have to appeal to really issues of motor output, and I mean I don't know. I feel you can't really make an absolute judgment here. I mean, theoretical neuroscience is pretty crude right now. I could go back to this example again of the central complex. You look at the data, you're measuring neuropil, and there's a blob, and the blob is shaking around, and just computing the average activity of that blob, basically the centroid of a blob moving on a circle, and we declare victory. And I agree you could declare victory, but someone else could say, well, but you're not really capturing the jittering of the fly as it moves towards a landmark. So I would say it's just an opinion completely that, just to get one decimal place in getting the average motion, in calculating the output from an animal is very impressive today. I would declare victory. I would give tenure to anybody who does that.
Awesome. Okay, so
Yeah.
I don't know if Dr. Engert is back, but if you are, we'd like to give you an opportunity to finish your thoughts
Yeah.
if you're on.
Yeah, no, I'm back. Sorry about that. No, I think actually, I'm not sure, I didn't hear what David said at the beginning, but I heard something about motor output is key, and I completely agree with that. I think that's the purpose ultimately of the brain, is to generate adaptive motor output. And ultimately, that is, I think, what needs predicting. Of course, I think it's not just the individual motor movement of any animal. What one has to predict is the motor output at many, many different spatial and behavioral and temporal scales, right? So if one can do that, is predict sort of the gist of the motor, the adaptive motor output, then I agree with David. Then we can declare victory. So the stochastic behavior of the animal that makes adaptive sense, if we can generate that, predict that, or simulate that, then I think yes, victory can be declared. And again, I think one way of generating these predictions is not just with algorithms on paper. I think the way we can predict is by running a network. That's a term I learned from David, right? If you generate a network, you hypothesize what it looks like, and then you run it in a computer, you add the actuators, the muscles, and you have an animal that actually behaves in a virtual world. And I think that is, to me, the best prediction that one can generate, and that we can compare now with the reality at the stochastic level. Over.
So, yeah, I agree. I seem to remember discussing this about 3 years ago in Washington, back when people actually took jet planes to go to meetings. You know, I mean, the problem is, particularly as a theorist, you get greedy, and you don't want just a computer output that tends to match. You'd like to, in a perfect world, get something simplified, some equations that talk to you, that maybe give you the voice, give you the motion in ways you could see, some simplification. Just like I'm trying to...
But that's the question, right? I think, do these exist, these simplifications, do they exist, or are we chasing a ghost here, right? Is the ultimate answer, is it just going to be a complex network that we can run and that will generate the same outputs, and that is just really complicated? I mean, it's a little bit like the theory of a cell, right? I don't think we have a simplified theory of how a cell works, yeah? But I don't think we are lacking it either. What do you think, David?
No, I agree with you. You have many processes in parallel with similar importance, and you just can't you can't knock it down to some.
Yeah, it doesn't compress
view.
Anymore.
Yeah.
There's no principal component that describes it all, or a simplified equation like the Maxwell equations, right, or the Navier-Stokes equation. I don't think that exists for the brain. I don't know, but I'd be skeptical.
Yeah. Okay. So I'm hopeful, but
good.
This is wonderful. So what I want to do is ask one more question from the audience to wrap up, and then we'll wrap up the discussion for the afternoon. So the last question is: won't any general theory of learning, for example, outside the context of the innate drives to preferentially learn certain motor actions, inherently take us down the wrong road? In other words, should the brain initiative be more comparative?
Can you repeat that, please? Wait, no. Can you repeat? I don't understand the question.
Absolutely. So I'll repeat the question. The question is, won't any general theory of learning, for example, outside the context of the innate drives to preferentially learn certain motor actions, inherently take us down the wrong road? In other words, should the brain initiative be more comparative?
Okay.
But comparative,
David.
you mean, just understand, comparative in terms of learning paradigms, or comparative in terms of animals?
I believe the context here is in animals based on the person who submitted the question.
Yeah, I mean that's a great point. I mean, there was a time when people would say you choose the animal to solve the problem that's most interesting. If it's a speech issue, you choose a songbird. If it's a manipulation issue, maybe it's a New World monkey. If it's an auditory issue, it's an owl, et cetera, et cetera. You really try to pick the right species, and we've gone away from that, and we've gone into mice and Drosophila and zebrafish, and maybe I'm missing something. And
Worm.
I think the worm. Yeah, sorry, it's
Yeah.
another group that I've managed to anger today. But the worm has its great capabilities in terms of molecular tools and the speed with which you could access things. But I really believe in the comparative approach. I mean, this gets banged into me all the time by my colleague Harvey Carton. These are nervous systems. These are, in principle, whatever the circuit is that's developed in one animal, that circuit or an analogous circuit could develop in a different animal. It's all giving us clues about the nervous system. I think we're pretty. I think we have to be honest with ourselves. Neuroscience is fascinating. Obviously, Florian, you, everybody at the NIH, we've devoted our lives to it, but it's a pretty crude subject at some level. And the more breadth of experimentation we do, and the more we could explain with our models, the more progress we'll make. So I would, I personally, even though we're not doing this per se at the moment, I put a big plug in for the comparative approach. Florian, what do you say?
No, completely agree. I don't really understand the dichotomy in the question. I don't think there's actually either we have a general learning model or we take the comparative approach. I think we sort of can do both. No, I don't also know what the general learning model, the learning theory, what that really means. I think that there is sort of a fundamental concepts in learning, like reinforcement learning or classical conditioning or sort of, I don't know, associative learning. And I think that's a good idea, yes, to try to conceptualize how learning works, to think about innate drives, about reinforcers, to think about how memories get written, how they get stored stably for a long time, how they get recalled. I think all of this requires a sort of conceptual framework. But I think the way you apply it and test it is by looking at as many different animals as you can. Like what David said, right? If you want to do interoral time difference comparison, you look in barn owls, and if you songbird learning is zebra finches and so forth. So I think that this is not an either or. I think having conceptual theories about learning, how it should work, is useful, is almost required. But in order to apply it, I think one almost has to go to a comparative approach, because that's how you learn about general principles, right? You can't just study a mouse or just a larval zebrafish. You have to, if you think you understand how something works, you have to test it in a broad range of contexts, of conditions, and therefore in different species. Over.
I think. Oh, go ahead.
No, I was saying I think we agree. That's last.
Okay, awesome. Well, that's a wonderful note then for me to say that we're going to go ahead and wrap up the debate. We've gone for the full hour period. I want to end this debate by saying I want to offer the profound gratitude of not only myself but my colleagues at the National Institutes of Health and the broader community for Dr. Engert and Dr. Kleinfeld, your willingness to participate in this and to do this as a dialogue and to talk about areas where there's differences. I think it's a wonderful, stimulating thing, especially for junior scientists, to help them think about what some of the next steps might be to push the field forward. I certainly learned a lot. I'm sure others have as well. So thank you both so much for your willingness to do this.
Oh, Mohammed, thank you so much. Yeah, no, thank you, not just for allowing us to discuss, but also for reconnecting David and myself. I've thoroughly missed talking to David and discussing science with him, and this just gave me a a really wonderful opportunity. And this was awesome.
Yeah, thank you. Also, I always enjoy chatting with Florian. We did the imaging course together many years ago, and I have to say this venue was much safer because one day Florian said, "Let's swim from Banbury to Cold Spring Harbor," and timed it perfectly as the tide was going out. And
but we survived. We survived.
We survived.
Makes you harder.
Wonderful. Well, I for one feel very optimistic that between sharp minds that have engaged in this debate, as well as several of the others we heard from earlier today, I think whatever the answer is, someone who's been on a call or given a presentation today will help us get to the answer, the bottom of it. So thank you both again for your time.
Okay, thank you.
Okay, over and out. Thank you, everyone, and greetings to the audience.
Yeah. Thanks to the audience for your participation.
Yeah.
Okay, bye-bye, Mohammed. Thanks so much for inviting us. And
Thank you.
okay, thank you, Jim and Karen and Christina. Okay, and Susan. Okay, thank you.
Bye-bye.